This video will demonstrate the steps required to design and order all necessary reagents for creating an insertion or deletion in a target gene using the Invitrogen TrueDesign Genome Editor. After logging into the Thermo Fisher Connect platform and accessing the TrueDesign Genome Editor, you have a number of experimental options to choose from. For this workflow, we will choose Insertion, which allows for inserting up to 30 base pairs of new DNA. Click Next. On the next screen, you'll see 5 species to choose from. Select the host organism for your experiment and click Next. You'll then select Gene Symbol Entree ID as input for how to identify your gene of interest. The other available options will use your input to navigate to the nearest transcript in the selected genome. Click Next. When you begin typing in the gene identifier box, a filtered drop-down list will appear. Select your gene of interest from the list and click Search Gene. The next screen will display all of the protein coding transcripts for your selected gene. If there is more than one displayed and you aren't sure which one to select, you can easily click the Transcript ID hyperlink to be taken to the NCBI website where you can visualize the transcript maps. Once you've made a selection, click Edit to proceed to the Sequence Editor view. The Sequence Editor will update to display the entire transcript's topology along the top of the screen with a zoomed-in sequence level view below. Use these views to navigate to the region where you will create your edit. In the Sequence Editor, click on the desired insertion location as you might in a text document. Enter the DNA sequence to be inserted and click Insert. The view will update to display the inserted bases highlighted in blue, and an edit list will appear to indicate the type of edit and its position. You can undo the change by clicking the blue Undo arrow in the edit list. At this point, you also have the option to introduce a second edit within a 30 base pair region of the insertion. For this example, I'll choose a deletion. This combination of an insertion and a deletion, when designed correctly, will replace a portion of your gene's coding region. Click on the Sequence Editor and select bases just like you would in a text document. When you are done, click Delete. The Sequence Editor view will update with the red arrow to indicate the region where the deletion will be, and the edit list will now indicate both your insertion and your deletion. When you're happy with the changes, click Design. This step might take a few moments while the software searches the region for available PAM sites and assesses the guide RNA designs for efficiency and specificity. When complete, you'll see a table with 2 tabs, CRISPR and TALEN targets. Each row in the CRISPR table displays a CRISPR guide RNA design, its PAM site, its score, which is a weighted score for efficiency and specificity, the number of predicted off-targets, the edit site distance from the CRISPR cut site, and the DNA oligo required that's as a knock-in donor template for your genomic edits. The single-stranded DNA donor is shown on top and the repaired DNA strand and amino acid sequence below. Green checkboxes indicate recommended guide RNAs due to score and proximity to the insertion site. Visualize the target sequence location on the Sequence Editor display by clicking anywhere in the row. To view the TALEN targets, click on the TALEN tab of the table, and similar information will be displayed for each TALEN pair. To select one or more guide RNAs or TALEN pairs to add to your experiment, use the checkboxes in the table and click Summary. The summary page will display all of the reagents needed for your gene editing experiment and give you the opportunity to add additional products to complete your workflow. For an insertion or deletion editing experiment, the guide RNAs and donor oligos selected in the design step are automatically included. For an experiment like this one, the tool will also add TrueCut Cas9 protein and lipofectamine CRISPR-Max Cas9 transfection reagent. These are added to your experiment by default but can be deselected from the product summary area, or a different quantity may be selected from the drop-down list. You may add additional items such as sequencing primers or positive and negative experimental controls. When you've completed your product selections, click Add to Cart for easy one-step ordering of all selected reagents. You may additionally download a detailed report of your experiment by clicking Export Results and Protocol.